Hello everyone and welcome. I'm Elahe and I would like to thank you for joining our webinar on uncovering pancreatic cancer biology with single-cell sequencing. Before we begin, please take a moment to review our disclaimer. Now let's take a look at our today's agenda. I will start by highlighting why sample preparation is a critical step that directly influences your experimental outcomes. In the second part, Dr. Alvaro Curiel Garcia will take over to share his findings in pancreatic cancer research. He will give you in-depth insights into pancreatic cancer biology, demonstrate how Miltenyi products enhance his workflow, and explain how to analyze and interpret resulting data in a real-world research context. Finally, we will wrap up with a Q&A session to answer all of your questions. Now, let's start with the first topic. So when we think about why sample preparation matters, it really comes down to one simple equation. High-quality samples lead to reliable and reproducible results. No matter how advanced your downstream analysis is, if your starting material isn't prepared properly, your result will be compromised. But preparing samples comes with challenges, and I'm sure many of you have experienced some of them. Issues with sample viability, reproducibility, and the time it takes to get samples ready. These challenges can slow research progress and sometimes even limit what is possible. This is exactly where Miltenyi Biotec comes in. Our mission is to bring standardization, automation, and ease of use to sample preparation to overcome all mentioned challenges. So we offer innovative tools and solutions that help at every stage of the process, from proper storage to dissociation, cleaning up unwanted materials, and subsequently ensuring success in your downstream applications. So the key takeaway here is that sample preparation is the foundation for reliable results. And with the right solutions in place, you can overcome these common challenges and focus on generating high-quality data. And now let's start at the beginning, from sample storage. This is where the sample journey begins. For many of you, especially when working with human materials, samples are collected in the operating room and then transferred or shipped to a research lab. Whether the transfer is short or long, it's crucial to preserve cell integrity to avoid changes in viability or gene expression. So we provide 3 different solutions for different conditions. Max Tissue Storage Solution is designed to store tissue samples or organs for short time, for example, for shipping or transferring to another lab. So you can keep the sample at 4 degrees for up to 72 hours without losing cellular composition or viability as shown in the data, and importantly, without inducing stress-related gene expression. But if you're working with a cell suspension, we offer the Max Cell Storage Solution as shown in the data, which maintains viability and cellular composition unchanged for up to 72 hours at 4 degrees. And finally, if you want to store your samples for longer time, we have the Max Freezing Solution, which is ideal for storing both cells and tissues at -80 degrees, and it ensures high cell recovery and viability. So we also have more supporting data available for each of these solutions, and you can find all details on our website. So once your sample is ready, the next step is to dissociate it into single cells or nuclei. The GentleMax Tissue Dissociator has been transforming this process for more than 15 years. As it's most commonly used for isolating light cells from fresh tissue, it can also be used to homogenize tissues for the isolation of biomolecules such as RNA or proteins. GentleMax technology combines mechanical dissociation with enzymatical digestion using tissue-specific enzymes in a single device and greatly reduces time and manual effort and ensures consistent and reliable results. Moreover, it's also highly effective for automated isolation of nuclei from frozen samples, a process that requires only mechanical disruption in combination with our nuclei extraction buffer. The GentleMax technology consists of three key components that enable standardized and automated tissue dissociation and homogenization. The first component is GentleMax tubes. They are specifically designed for optimized mechanical disaggregation. Two tube formats are available: C tubes with purple caps designed for cellular application to efficiently dissociate tissue into viable single-cell suspension, and M tubes with orange caps optimized for the homogenization required for downstream molecular analysis. The second component, the tissue-specific dissociation kits, provide validated tissue-optimized protocols with purified enzyme mixes that ensure high yields of viable single cells from virtually any tissue type. These components are designed to work seamlessly with the GentleMax octodissociator with heaters, an automated instrument that can process up to 8 samples simultaneously or independently. By combining mechanical dissociation with enzymatical digestion, the system provides a user-friendly, standardized, fast, and highly reproducible sample preparation workflow. So the success of GentleMax technology is reflected in the global adaptation. This graph, showing more than 8,500 publications, tells a powerful story about the adaptation and validation of GentleMax technology within the scientific community. The rapid and sustained increase in publications citing GentleMax is a strong indicator that researchers worldwide are increasingly relying on this platform for their tissue dissociation needs. This instrument, however, offers a range of powerful features designed for both flexibility and efficiency. Its intuitive software provides clear, step-by-step guidance with options to filter programs and pause and resume the experiment. It offers temperature control. Samples can be processed either at room temperature or 37 degrees or even low temperature using the GentleMax OctoCooler. As mentioned before, the system supports the processing of up to 8 samples, either simultaneously or independently, giving you maximum flexibility in your workflow. It also comes with more than 80 predefined programs and the ability to create customized protocol tailored to your specific needs. Finally, the system also supports ex vivo perfusion, enabling the gentle isolation of fragile cell types such as hepatocyte or cardiomyocytes. Let's take a closer look at the tissue dissociation kits. These kits provide a standardized way to dissociate tissues with over 25 different options available to suit various tissue types and research needs. Each kit is ready to use, containing high-activity enzyme and optimized buffers that help preserving surface epitopes, an essential feature for reliable downstream application. They also offer lot-to-lot consistency, ensuring your experiments are reproducible. And with pre-tested protocol, the kits save you time and simplify your workflow significantly. On the right side of this slide, you can see examples of some tissue-specific dissociation kits we offer, and we have also more that you can find them on our website. So following tissue dissociation, now it's essential to clean up the cell suspension to remove unwanted materials. We offer a range of solutions designed for this purpose: innovative filters and cell strainer with different mesh sizes, along with specialized cleaning reagent. These tools help to prepare your sample for downstream application effectively. And you can just simply removing your cell aggregates, debris, and other unwanted components such as myelin, dead cells, erythrocyte, or organelles. So now let's summarize the complete workflow and its implementation for single-cell sequencing. Fresh tissue can first be stored in an appropriate tissue storage solution. Next, dissociation is performed using the GentleMax dissociator together with the tissue-specific dissociation kit and GentleMax CTU. After dissociation, the samples are cleaned to remove debris and unwanted material. Target cells are then isolated, followed by a quality control step to ensure that the resulting cell suspension meets the necessary criteria for successful downstream application, such as single-cell sequencing, flow cytometry, or any other analysis. This is the general workflow that can be applied to various tissue types. Depending on your research goal, some adjustment may be required. Later on, Alvaro will show how he has implemented this workflow in his research. Now, moving on to nuclei extraction and enrichment, another really important and widely used application in the field. Nuclei preparation is a critical step for downstream genomic analysis, such as single-nucleus RNA sequencing. Compared to single-cell RNA sequencing, single-nucleus RNA sequencing offers several advantages. It enables the analysis of challenging tissues such as brain, liver, or muscle. It works effectively with frozen tissue samples and allows direct access to the nuclear transcriptome. To ensure high-quality data and reliable results, it's essential to use an optimized and standardized workflow for nuclei preparation. So here is the overview of our workflow to isolate nuclei suitable for single-nucleus RNA sequencing and consists of two main parts. The first part is nuclei extraction to release nuclei from tissue. The workflow is straightforward, rapid, and highly reproducible. Fresh or frozen tissue samples are placed into a GentleMax CTU containing the nuclei extraction buffer. The GentleMax dissociator comes with a predefined nuclei extraction program. If this program isn't available on your device, please make sure the software is up to date or contact technical support for assistance. Throughout the extraction, the GentleMax octocooler maintains a low temperature, which is critical for preserving nuclei integrity. The entire extraction process is extremely fast and taking only about 5 to 10 minutes. After mechanical dissociation, the resulting suspension is filtered through a 70 micrometer smart strainer to remove debris and aggregates. Validation data demonstrate that this extraction protocol performs consistently across different tissue types and conditions, including fresh, snap frozen, and OCT embedded samples. So following extraction, the nuclei suspension may still contain residual debris. So the next main step is nuclei enrichment. This is done using antinucleus microbeads and very well-known max magnetic separation technology. This enrichment process is simple, doesn't require a specialized instrument, and can be completed in under 30 minutes. After extraction, the sample is spun down and nuclei are counted, resuspended with the antinucleus microbeads, and then passed through an LS column within a magnetic field to isolate pure nuclei. So once enrichment is completed, quality control can be performed, for example, using max quant analyzer to assess nuclei quality and concentration. Overall, the complete workflow from tissue sample dissociation to high purity nuclei ready for sequencing can be completed in less than 1 hour. So now I want to really emphasize that this enrichment step is crucial. As an example, we used frozen mouse brain tissue, and as shown in the data, debris levels can be quite high in frozen samples, before enrichment. In fact, nuclei purity can be as low as 5%. However, following enrichment, purity increases significantly to 87%, which makes the following single-nucleus sequencing much more efficient and reliable. Furthermore, the enriched nuclei exhibits preserved integrity, appearing round or slightly oval with a smooth, intact surface, indicating that the enrichment process maintains nuclear quality. So with this slide, I would like to thank you all for your attention. Together, we have explored how our solutions empower both single-cell and single-nucleus research. Now it's time to see this in action. I am pleased to hand it over to Alvaro, who has hands-on experience with both workflows. In this webinar, he will mainly focus on best practice for sample preparation for single-cell sequencing and share real-world example of how he has implemented these tools to generate meaningful insights and advance single-cell research. Thank you.
Hello everyone, and thank you so much for joining today's webinar. My name is Álvaro Curiel García. I'm an Associate Research Scientist at Columbia University Irving Medical Center in Dr. Ken Olive's laboratory. We focus on pancreatic cancer biology, and we make heavy use of single-cell RNA sequencing to study tumor heterogeneity, the tumor microenvironment, and therapeutic response. The goal of today's talk is to share with you the protocol we have adapted using Miltenyi's reagent for generating high-quality single-cell suspensions from pancreatic tumors. As you may know, pancreatic tumors are extremely challenging tissues. They are dense, fibrotic, and often necrotic. Getting viable cells out of them is not straightforward. Today, I will show you the workflow we use, highlighting critical points where small details make a big difference, and share how this protocol has enabled us to generate high-quality single-cell data from mouse models, including a publication, and I will share at the end of the presentation a small tips to help isolating single cells from human patient tumors. To begin, these are the essential supplies you will need. In addition to the obvious items like the tumor sample, the sterile scissors, surgical pipettes, tips, and conical tubes, you will also need a 70-micron cell stainer. This is the perfect pore size for pancreatic tissue, as it removes clumps but still allows single cells through without much stress. Then you will need the Gentle Max C-tubes as well as the Gentle Max Onto dissociator. This instrument is absolutely central. It standardizes the mechanical dissociation process, which means we no longer rely on manual chopping or inconsistent digestion. Finally, you will need ice and ice-cold sterile DMEM ready. Pancreatic tumors can release a lot of enzymes and reactive species once they are disrupted. So keeping everything cold is the best way to protect RNA integrity. Consistency is critical when you want reproducible sequencing data. Now, for the enzymatic part of the protocol, we use Miltenyi's optimized digestion cocktail. This consists of enzymes D, enzymes R, and enzymes A, combined with DNAs and a trypsin inhibitor. Each of these plays a specific role. Enzyme D is mainly collagenase-based, breaking down extracellular matrix. Enzyme R complements this by digesting other stomach proteins. Enzyme A is a more general protease. DNase is very important. When cells die during dissociation, they release DNA, which makes the suspension viscous and clumpy. DNase keeps the suspension fluid. Finally, the trypsin inhibitor, which prevents overdigestion, protecting surface proteins that are crucial for downstream analysis like flow cytometry or cell hashing. This exact mix is something we found to work best after testing multiple options. Here you can see the recipe we prepared for each individual tumor. We precisely measure small microliter amounts of each enzyme, then add trypsin inhibitor and DNase, and bring the total volume to 2.5 milliliters with the MEM containing 5% FBS. One point I want to stress is, make this fresh every time. Don't prepare the cocktail in bulk and freeze it. Enzyme activity can be lost, and variability will increase. Also, have it prepared before you even begin dissecting the mouse. Once the tissue is out, the clock is ticking. The faster you get the tumor into the digestion mixture on ice, the higher the viability. Now, let me make you through the actual protocol step by step. We will begin by making up the digestion buffer prior to sacrificing the mouse. Have everything chilled on ice and ready. Sterile scissors. Use soap, water, ethanol, and then flame them and place on a sterile surface. Sacrifice the mouse and record weight of the total tumor. Mince the tumor on ice in digestion buffer into 2 to 4 mm size pieces. 3-4 pieces are more than enough at this step. Performing them in gentle maxi tube. Tightly close the C-tube and attach upside down to the octo-dissociator with scissors. Run the 37C MTDK2 program. This combines mechanical disruption with precise heating for optimal enzyme activity. Sometimes the first round doesn't digest everything. If that's the case, remove the supernatant, keep it aside, and add fresh DMEM to the remaining tissue. Then run the MIMP tumor 01 Miltenyi program. Then recombine everything. Remove the supernatant and add to the 50 ml conical tube. Add fresh DMEM solution to the C-tube with the remaining tissue and perform a source centrifugation step to collect all sample material at the bottom of the C-tube. The more material, the better. Then resuspend the sample and apply cell suspension to the 70 micron cell strainer placed on a 50 ml conical tube which has been pre-wet with chilled DMEM. Incubate the cells on a strainer with 0.25% trypsin EDTA for 5 minutes. This extra step can release additional viable cells from clamps. Then wash the cell strainer with another 15 ml DMEM and centrifuge the cell suspension for 7 minutes. Aspirate the supernatant manually and keep it in a separate tube. If you believe you have a very small amount of cells, there's an extra step you can do. Spin down the supernatant for 10 minutes in a second time. Then wash the bottom of the tube with a small volume of DMEM and combine with other cell suspension. Then you want to remove the erythrocytes from the sample, so perform the red blood cell lysis protocol using your favorite red blood cell lysis reagent. In this case, use the 5 ml conical tubes since they have a lower surface and will help you out spinning down all the cells later. Up to 3 washes with the resuspension protocol, which is a PBS and 0.01% of BSA. If you barely see the pellet, make only 1 wash and check the variability. Once the single cell suspension is ready, we check the variability. Ideally, we aim for above 80%. In pancreatic tumors, this is actually quite good because the tissue is so fibrotic and harsh. What makes the difference here is really attention to the details. Again, keeping things cold, minimizing mechanical stress, and handling every function carefully. These are the same principles that apply across many tissues, but in pancreatic cancer they are especially critical. Here, I would like to show you one example of how we have applied this protocol in our research. This is a study recently published by our group in Cancer Discovery, where we use the tumor pancreatic from genetically engineered mouse models of pancreatic ductal adenocarcinoma. In this paper, we show how paracrine signaling cascade involving the sonic hedgehog Wind a VEGF regulate tumor angiogenesis and progression. To generate the single-cell RNA sequencing data that supported these findings, we relied directly on the Miltenyi mouse tumor dissociation kit. So the protocol I've just described is not only theoretical. It's been validated in a peer-reviewed work and has enabled us to answer important biological questions about how pancreatic tumors communicate their environment. Here are some quality control metrics from one of the experiments. We obtained over 8,400 cells from a single tumor preparation, with an average of almost 40,000 reads per cell and around 700 genes detected per cell. The sequencing saturation was about 36%, and both UMI and barcode variability were near 100%. What these tumors tell us is that the dissociation protocol yields suspensions that are not only viable, but also give us excellent transcriptome coverage, one sequence on the 10x platform. In other words, the method is robust enough to consistently produce high-quality data suitable for downstream analysis. Here we see the results of single-cell clustering visualization in UMAP space. On the left, you can see the different cellular compartments present in the pancreatic tumor: myeloid cells, lymphoid cells, fibroblasts, epithelial cells, and endothelial cells. This demonstrates that our dissociation method captures a broad range of cell types, not just tumor cells, which is crucial for understanding the tumor microenvironment. On the right, we overlay treatment conditions: vehicle versus IPA 926, a smooth inhibitor targeting the HOT signaling pathway. You can see that treatment-dependent shift in cell populations, highlighting how single-cell approaches allow us to capture the heterogeneity of responses within the tumor. Finally, here is an example of the type of analysis enabled by this protocol. This dot shows differential expression of the HOT pathway genes in different cell compartments after treatment with IPA 926 compared to the vehicle. You can see that the genes G2, G3, and PATS1 are significantly downregulated, particularly in fibroblasts and endothelial cells. This supports our model that HOT signaling pathway plays a central role in regulating stromal tumor interaction in pancreatic cancer. Again, I want to emphasize that any of this data could be possible without starting from a reliable, reproducible method to dissociate very tough tumor tissue into variable single-cell suspensions. Let's now move to the human samples. As you might expect, human pancreatic tumors are even more variable than mouse tumors. They can be fibrotic, sometimes necrotic, and often we have less material to start with. For this protocol, we have the same reagents. The only difference is that we change the enzyme D from the mouse pancreatic tumor protocol to the enzyme H. The remaining parts are the same, including the quantities. In this case, we are also switching the program from the octo dissociator to the 37H TDK3 program. By adapting this protocol, we've been able to successfully generate single-cell suspensions from multiple patient-derived samples. These suspensions go directly into our 10x genomic pipeline for RNA sequencing, and the data quality has been excellent: high complexity libraries, low mitochondrial read fractions, and a strong representation of both tumor and stromal cell populations. I want to conclude by thanking the people who made this possible, as well as my laboratory, Dr. Ken Olive, the colleagues who optimized and tested this workflow, and all of our collaborators. Of course, Miltenyi for providing reagents and technology that really make this possible in such challenging tissue types. Thank you all for your attention, and I hope this presentation not only gives you a protocol you can apply, but also some practical insight into the small detail that makes single-cell RNA sequencing experiment success or fail.